Hello and a very good day to our lecturer. In this video, we will be discussing about chromatography analysis for determination of carbohydrates in food products, for example, yogurts. The first one is introduction. Carbohydrates are a fundamental component of many foods, contributing to their nutritional value and energy content, as well as participating in many biological processes including cell recognition, development, interaction, and inflammation. High-performance liquid chromatography, also known as HPLC, is used for analyzing carbohydrates due to its high precision and ability to separate complex mixtures.
Next, we go for theory. The principle of HPLC, High Performance Liquid Chromatography. HPLC purification is a technique where the sample is injected into the high-pressure stream of a solvent through a column packed with a porous particle that is a stationary phase through which components migrate with variable migration rates and different interactions that are detected after exit from a column, and a chromatogram produced for identification and quantification. For example, we can look from determination of soluble carbohydrate in yogurts by high-performance liquid chromatography experiment. From the experiment, it described an HPLC procedure using a sugar pot 1 column at 85 degrees or 90 degrees and a refractive index detector with double distilled water as the mobile phase, which made it possible to simultaneously identify and quantify the sucrose, lactose, glucose, galactose, and fructose present in yogurt, and it shows good separation of these carbohydrates with resolution without interference from other components.
For the methodology, materials and methods, 10 units 125 to 150 gram each of 5 flavors of yogurt with the same code date were purchased at a local retail market. 2 brands of yogurt also were used. Samples then were blended in a laboratory blender. Dry matter content of each sample was calculated from weight loss after heating in a vacuum oven at 35 degrees Celsius to constant weight. For the preparation of carbohydrate extracts, samples of yogurt were weighed and ethanol water was added to a final ethanol concentration of 80%, and the mixture was boiled under reflux for 15 minutes. The mixture was cooled and filtered through a glass funnel. The residue was extracted twice and washed with 80% of ethanol. The extractive liquids were evaporated under vacuum at 40 degrees Celsius. Residues were dissolved in distilled water and extracted with ethyl ether to remove substances as colorants and fats that interfere with the carbohydrate analysis. The aqueous phase was reduced to dryness and dissolved with distilled water. The solution was purified and filtered. The analysis of soluble carbohydrates. The identification of soluble sugars was on thin layer chromatography with plates with silica gel impregnated with boric acid. Then identified and quantified sugars by HPLC, comparing the retention volumes with corresponding standards. For the apparatus, the high-performance liquid chromatograph was equipped with a pump with dual reciprocating piston heads held at 85 degrees Celsius or 90 degrees Celsius with a water column temperature control accessory, pre-column filter and differential refractometer detector, optical deflection type maintained at 30. The detector signal was recorded on the data module child recorder. As for the mobile phase, distilled water was filtered and degassed by immersion in an ultrasonic bath. As for the flow rates were 4 ml per minute and 5 ml per minute. As for the standard solutions, different amount of glucose, fructose, galactose, sucrose, and lactose were dissolved in distilled water. Then the solution was filtered. Quantification of each sugar was accomplished by comparing the peak height of the samples to that of the standard curve solution.
Next, I will present the example of result and discuss the result. The study showed that every yogurt sample had galactose and lactose. Glucose and sucrose were only found in the flavor yogurts, so no samples had fructose. Table 1 lists some important details about the sugar part 1 column used in the test. To measure the soluble sugars, high-performance liquid chromatography (HPLC) with special columns was used. This column separated glucose and galactose well, but not sucrose and lactose as effectively. Lactic acid can interfere with galactose measurements, so a biorate microgut anion/OH module was used to reduce this interference. However, this module also absorbs some sugars which might affect the results. The sugar part 1 column, which is an ion exchange type, did a good job of separating sucrose, lactose, glucose, galactose, and fructose using double distilled water. The analysis found that sucrose and lactose were much more common than glucose and galactose. It's recommended to test the sugar with different sample amounts for accurate results. Table 2 and 3 show the average result and standard deviation from 6 tests of the sugar content in 5 yogurt flavors. Galactose and glucose in yogurts come from the breakdown of lactose. The data show that glucose level were always lower than galactose level. This happened because yogurt bacteria, like lactic acid bacteria, use up glucose faster than galactose. These bacteria prefer glucose as their main energy source because of their metabolic processes. The chromatographic conditions described in this analysis are particularly good for measuring soluble sugars in fermented dairy products by HPLC. This method provides a reliable and accurate means to quantify these carbohydrates, ensuring that even minor components can be effectively separated and measured. This is important for understanding the carbohydrate composition of yogurts and can help in quality control and product development in the dairy industry.
As a conclusion, chromatography analysis for determination of carbohydrates in food products offers a precise and reliable method for identifying and quantifying various sugars. Accurate determination of carbohydrates content is vital for nutritional labeling, quality control, and dietary management.